Hello, my name is Jason Keagy. I am an assistant research professor at Penn State University, and I'm going to tell you about work I did as a postdoc with Alison at the University of Illinois on the behavioral and neurogenomic effects of receiving parental care. So there's a number of ways that parents can influence their offspring. So they can influence them through the actual genes that they're giving their offspring. They can influence them through the developmental environment of the embryo, either in the egg or the uterus. And then the category that I'm going to focus on for this talk is that parents can give care to their offspring after birth, and that can have effects. But most of this literature on parental care effects after birth has been on moms. And this is focused on things like maternal style, so the way that mothers are behaving towards their offspring, teaching of the offspring, or protection of the offspring from predators. And what Alison Bell has been doing is developing the stickleback model to explore how paternal care can affect offspring as a comparison to the much larger literature on maternal care. We had a number of different research questions. First, how does paternal care affect offspring behavior? Second, through what genetic mechanism does this occur? So we focused on two, the brain gene expression. So um, think about like whether genes are on or off, or how high they're turned on and off. Um, and then we also looked at chromatin accessibility, or you can think of this as gene accessibility, whether a gene is actually available for transcription. And then we were interested in whether there were any sex-specific effects of paternal care, because female and male sticklebacks have very different sex roles as adults. So we had the following experimental design to create offspring with different levels of care. Males would build a nest and then mate with a female, so they get eggs. And then in one case, the male was allowed to stay in that tank and take care of his babies for the same amount of time he would in the wild. And these were called father-reared offspring. In the other case, we took the eggs out of the nest and put them in this bubbler system and did minimal intervention. And these were called orphaned offspring. And then we took these males and allowed them to build a new nest in a new tank, mate again, get eggs, and then that second clutch received the alternative treatment. So males in all cases had one clutch that received paternal care from him, and then another clutch that did not receive parental care. And we had 16 different families in this experiment. We then did a number of experiments. I'm going to focus on one here in this talk. Basically, we let these offspring grow up, and just before they were sexually mature, so around 7 months old, so they still have undifferentiated gonads at this point, we presented them with a situation where fish were individually placed in an open field assay. You can imagine that stickleback would have been a little smaller than this red dot here. They would explore this environment, and then a predator, a model predator, chased them around in a circle, and then we looked at how they behaved after that exciting encounter. And then 1 hour after that, we took their brains, and half of the brain we used processed for gene expression study, and half of the brain we processed for that gene accessibility study that I'll tell you about. We also had what we call baseline controls, or you can think of them as circadian controls. So for each fish that went in here, within half an hour of that fish's getting its brain dissected, we took a full sibling from its tank that hadn't had any of these crazy experiences, and also took its brain and processed it for these two different studies. We had roughly 60 fish that went through the predator treatment for each paternal care treatment, and the same number of fish as baseline controls. So let's talk about behavior. Here on the x-axis, I have the time period. So fish actually, and this is a binge, so fish actually went in for 10 minutes, then they were attacked, and then they had another 10 minutes. On the y-axis is the proportion of time they spent under the plant. So these fish would be those that hid the entire time, and fish down here would have hidden none at all. We generally see, like you might expect, after getting attacked, fish hid more. We also, you'll notice that these circles here, these are the orphaned, are always above these triangles. the father redfish. So it seems like fish that received care were hiding less. But it's not that simple. We have a three-way interaction between paternal care, time, and sex. And that's because the strongest effects were seen after the attack. You'll see these lines have separated. And then the really biggest effect was between females. So the females were really different, especially after the attack, with orphan fish hiding quite a bit more than the father redfish. We see not really much of a difference between the male fish. Let's talk about gene expression. These are called volcano plots because they kind of look like volcanoes spewing lava. On the x-axis is log2 fold change. The way you can think of this is this one here means that genes here are expressed doubly in the fish that in this case were orphaned versus the fish that were received paternal care. And then we see this is color coded to be, these are uh, anything colored is statistically significant after a false discovery rate. This y-axis, the way you can interpret this is that these, as you go up, genes are much more statistically significant. And then they're color coded to correspond to genes that are red, were more expressed or higher expressed in the father redfish. Genes in blue are more expressed in the orphan fish. We see 800 genes that were actually differentially expressed between these two groups, um, and quite a few more were differentially expressed in the father redfish, such that they had higher expression than orphan fish. We see very large sex differences, actually a similar number of genes involved. And we actually captured some genes that were directly affected by the experience of being in that tank and attacked by a predator. Now I'll talk about gene accessibility. So we actually only found large sex effects, but they were quite large. Yeah, we did not see any paternal care effects, however, although this study is much less power because of this is a much more expensive technique. So just to summarize, we saw that fish who received care were more bold after the predator attack, and this effect was most apparent in females. There were large parental care and sex effects on gene expression, and some gene expression differences from the predator exposure itself. And there were also large sex effects on gene accessibility. And what we'd like to do now is actually try to put these data sets together and actually look for links between behavior, gene expression, and gene accessibility. And so with that, I'd like to thank NIH for funding to Alison Bell that supported this research, as well as a whole bunch of undergraduates